And then I'm gonna hand it over to Dr. Chow for him to give us a prayer and start to speak. And then maybe toward the end, or depends on what Dr. Chow's want, we can allow for Q&A, and then we finish it up with a prayer. All right, so Dr. Herbert Chow has been a Christian since age 12. I don't know how old he is now, but it's a long time.
About 14.
About 14. He had been with our Reason to Believe community for a while now, but we have the privilege to invite him to talk about this topic because he's an expert in this area of COVID-19 vaccine. He graduated from Illinois University with a Ph.D. in Experimental Immunology, and had 30 plus years of experience in the business, including working for Johnson & Johnson and Abbott Labs at several positions, all in vaccine development and some biotech startup company. So without further ado, Herbert takes over.
All right. So maybe before we start, let's say a quick prayer.
Okay.
Father, guide us through the learning today on the COVID topic and guide our decision forward in this difficult time. Let us humble in front of you and learn the beautiful signs that you have bestowed in our immune system to fight pathogens. We want to claim the promise that if we first seek you and your righteousness, everything we ask for will be given to us. In Jesus' name, amen.
Amen.
So David set me up because he set up a high expectation. So I did have a few years, even though I'm 14, but I did have a few years working in the vaccine business. In fact, most people think the vaccine is only for infectious disease, but in my world, we do vaccines for cancer, vaccines for even neurodegenerative diseases. So the vaccine goes a long way these days. So it's not a narrow definitions of bacteria and viruses. So let me share the screen. So today we're going to talk about SARS and COVID-2 infections, and I give you a rundown on the COVID vaccines on the market. And obviously, you're going to hear that there are a lot more vaccines coming beyond the 3 that are now approved in the United States. So I have to thank David and the committee gave me the opportunity, not only 1 session, but 2 sessions. So this month, we're going to focus today just on the SARS-CoV. And next week's topic, we're going to go from the micro world to a world that can only measure with light years. So we want to talk about the universe. And what I'm hoping to accomplish in the 2 sessions is to refocus you that there is intelligent design through the lens of microbiology and cosmology. So if you allow me for two minutes of infomercial, I'm going to tell you what we're going to talk about next month, and then we come back to the SARS-CoV. So you all know that in Genesis 1, God made the claim that he is not only the only God, but he's also the creator of the universe. And for thousands of years, the world tried to take the title back away from him. And the battle has intensified since 1930s, I guess, when George Lemaitre, he's a well-known astrophysicist and a contemporary scientist with Einstein, and also a practicing Catholic priest. And he was the one who proposed the theory of Big Bang. Oops. So since then, the science of Big Bang has mushroomed into a well-organized, magnificent, unified models with the fancy names of lambda codot meta model. And this has really now solidified the entire world of precision cosmology. And it's a beautiful model. And it has the support of multiple Nobel Prizes, as well as 70 years of NASA space science. So it's a great science breakthrough in terms of understanding the history and the origins of the universe and how it is made. So what we're going to do next time is, if you allow me, to walk you through the model and take the key findings of the models and reference back to Genesis 1. But by the way, if you take all the words from the Bible, there won't be more than 350 words to talk about creations of the universe. So we're going to do the referencing between what's in Genesis 1 and this model, and look at the science behind it. And what I want to do is ask two very bold questions. One, is God still the creator of the universe? And two, was he in full control of the creation science from day one? So those are the two questions we're going to ask. And you're going to ask me, well, I don't need to know that, because all I have to do is I have faith. That's what I've been working on that model for my entire adult Christian life, until 3 years ago. An event changed my thinking. So that's what I want to share with you next time. And hopefully we get to talk about this. So without any further delay, let me focus then to the topic we want to talk about today. So whenever I talk about COVID-19, I'm talking about the disease. Whoops, hang on. I hate that when this happens. So the COVID-19 is the disease that was caused by the virus SARS-CoV-2. SARS stands for Severe Acute Respiratory Syndrome. CoV is the name coronavirus. 2 means there is a 1. 1 showed up year 2000 and 2001, which has a name of SARS, and killed about, well, I guess the fatality rate of that virus is about 10%. Compared to the current virus, it's only 1.5%. So what we have today is a mild sister virus of the day past. And there's another sister virus that showed up 10 years ago called MERS, M-E-R-S, and that came directly from camel, and that even had a larger fatality rate of about 30%. This is the electron microgram of the virus, and you can actually see that the spikes is readily observable in this electron microgram. And this is a cartoon representation. You must have seen it many times. And this is the spike, right? And then if you really represent it in terms of the spaghettis, are actually the protein strain. If you unravel this and put it on, you actually see that these spaghettis wind around. And this represents a total amino acid count of about 1,273 amino acids. So I'll show you all that in a little while. So what I want to do today is, you can't talk about the vaccines and understand whether the vaccine is good or bad, not until you understand how the variants and how the mutation comes about, and what forces are shaping the vaccines, or not the vaccine, but the development of the variants. And I have to tell you that the pandemic environment we have, it's totally different from the pandemic what we have in 1918. Obviously, the population is much larger now. Back then, you don't have to do social distancing because you don't have a lot of space in between. All you have to do is don't go downtown and just stay in your house, which is many miles out from downtown. The pandemic will die down. A lot of people die, but it's a lot easier to control pandemic back then. But of course, the medical practices were pretty lousy back then. That's why a lot of people died. Today, because of the populations, doing social distancing is difficulty, let alone that we can now zip all over the world. You can go to Paris and back to San Diego. You can get onto a cruise ship, and we see this kind of a problem last year. Once you get on, some of them couldn't get off because you have to be quarantined if there's an outbreak of coronavirus in the on the cruise ship. We also practicing something that we never did, in 1918. It's called intense farming, because now we have so many more mouths to feed because of the populations. We do these massive intense farming of pigs and chickens because of the high density. If there's a spread of virus in the population, it just spread like Wi-Fi. And I'm going to show you today, actually, COVID-19 variant has a root in some of these intense farming. You probably haven't heard from the news, but I'm going to tell you that story. And of course, we have the global shifting in the weather pattern. Some of the birds, bats, mosquitoes that wasn't close to us before, now they are shifting. They're coming to you instead of you trying to avoid them. If you deforest and try to build homes around a cave that there was a swarm of bats, you are that much closer to diseases than your parents that living 100 years ago. So with all these things going on in the background, it is not too surprising that if you allow viruses to commingle, different strains or different mutant to commingle, they're going to mutate, because recombination is a way how they live. And I'm going to give you example of how intense farming actually precipitated to close proximity and allowed these changes to happen a lot more rapid today than yesterday. Allowed us, the virus, to get closer to human. It also, by circulating through the host, it allows them to sharpen their skill to find ways to enter our cells. And we'll see examples of that today. So it is no longer a theory now. So whatever all these things are happening, it's causing the virus to be more virulent. We see that every day. So, it's no longer people can, oh yeah, you're just talking about theory. No, we're seeing viruses getting more virulent, they spread faster, high infectivity, because now they have a lot more opportunity to hone their skills, not only within the human population, but within animal populations. On the other hand, we have the natural immunity that built into our biology that in equilibriums with the viruses. And we've been living with viruses for eons, right? And for a long time, we don't see all these aggressive behaviors of the viral particle. But as we build immunity, and the immunity that we're building with the natural infections are what I call broad spectrum immunity. Because if you look at the viruses have spike proteins on it. In addition to that, there are a lot more targets that it will stimulate our immunity. With the broad spectrum immunity, we have a better controls on the viruses. And in essence, we are in equilibrium for a long time. Now, we want to develop a vaccine. We also want to have vaccines that are broad spectrums to mimic what the biology is doing. In that way, we can control the virus equally well. If we don't do that, if we develop vaccines that are kind of wishy-washy, then we're creating a problem, because we are producing pressure for the viruses to evade the wishy-washy vaccines. The viruses are allowed to escape. And I'll show you what immune escape it's all about. And you hear all of these terms in the news. So, you need to know about it. And this side of the equation, I would not talk about. I'm going to leave it with the pros to talk about what vaccine is best for this particular virus. what the coverage of the spectrums it's appropriate, and how do we unfold the vaccine? Should we vaccinate everybody, or should we just vaccinate the vulnerable population? All of these have impacts on how we shape our relationship with the virus in the future. But be that as it may, we have now a very powerful virus vaccines on hand. And the questions we need to ask is it's not whether this is the best vaccines. It is a very strong vaccines. Whether it's the best vaccines, it's not the question we want to answer today. But giving those strong vaccines, we need to understand what are the risks and what is the reward, so that we can make informed decisions to take the vaccine and how to work with it. The vaccine, as of this moment, is probably the only powerful tool we have to break the grip away from the COVID. And we really need to stop the spread of the disease and stop the circulations of the virus among the human population. Because if you let it keep circulating, they will keep mutating. And that's the key message. So we're gonna make references to this diagram off and on tonight. Once we understand the vaccines, once we understand how the virus adapt to the pressure of the vaccines. And at the end, I'll come back to make comments of some of the do's and don'ts that we have to think about, how to modify our behavior or misbehavior. And with all that, understand the drivers, how the viral infections comes about, and how viruses adapt to the pressure of the immune systems to either continue to mutate or to stop. So what I really want to do is at the end, I'll give you the source of the informations that I draw upon, and none of these I get from news. So you're not going to hear me say Fox News said that, CNN say that. I really don't. All the articles I get, some of them are peer reviewed, some of them are government sanctioned. These are from qualified folks who knows what they're talking about. And I want you to have these so that you also, with some of the vocab that I'm going to give you, do your own research and fact check what I said, and don't just listen to the mass media tells you whatever they want to tell you. Right? Let me at this point also give my disclaimer. Even though David said that I work with Abbott, AbbVie, and J&J. I've been consulting for a lot of these small companies doing vaccines in the last 10 years. At this point, I don't own a single stock in Johnson & Johnson's or any of these vaccine companies. I'm not working with them. So I really have no reason to say something, whether it's good or to say something is bad. I'm just based on my knowledge and based on what I learned from the professional scientific community. I'm trying to give you my 2 cents, and hopefully it helps help all of us make decision and make better decision. I know some of you are professionals, that you know probably more than I do. So if you hear and see some of the things that I said, it's has error in it, please feel free to email me so I can get some of these things corrected. So what I want to talk to you are 3 areas. One is tell you a little bit about the virus itself. I want to tell you they are nothing but hitchhikers, the lock hackers, and the hijackers. The 3H foundation. They don't even qualify the 4H foundations. They're nothing more than that. And I want to tell you a little bit how they reinvented themselves to make themselves, and you hear variants all the time. It's actually some of the variants are now causing surge of infections in Europe. Italy is about to go into lockdown again. And I'm the variants come to United States a little bit later than UK and South Africa. In January, I would say they're no more than a few percentage. If you survey all the virus samples from patients, most of the viruses are not the UK variant and not the South African variant in time frame of January. At this moment, we are close to maybe 40, 45%. As it progressed through 50%, I don't know what's going to happen. All right. So as some of the states is now opening up for no mask, go back to normal life, we are in the background seeing these variants are now coming up the rank. I also want to impress you, these variants, it's a very interesting story. And after that, I want to tell you about the vaccines, how many are there, how they work, what's the common goal. And then I'll give you, how I see them and how they compare and what are the risks and what's the reward. And the third area, it's what I really want to talk to you about today, if we have time. It's how the immune system actually deal with these constant threat of pathogens. The variant is going to change regardless, but how is that our immune system can always be able to keep up? We're probably not ahead of the curve, but we are more or less half step behind the most baddest viruses that are mutating as of today. We are no more than a half step. So I want to impress you in terms of intelligent design. I can see the handprints of God in the designs of our immune system. So I'm excited to talk to you about it. We're dealing with something really small. If the diameter of your hair is 80,000 nanometer, the virus we're dealing with, it's about 50 to 120 nanometers. So it's like, 1,000 folds smaller. Bacteria is obviously bigger. This is really small stuff. And the next level down at about 2 nanometers, you'll find your DNA. And then go down to about 0.1 nanometer, you find an atom. Okay? So you want to know how big a gold atom, it's about this size. It's small. And this is your hair, right? Now, for me, I don't have as much hair, so I don't care. But I do care about the virus. Okay. So I'm going to give you a lot of words, a lot of names. I don't want you to remember these names. At the end of the day, you're not going to remember any of these names. But I want to show you diagrams, how things work together. So hear the story, but don't remember these names like, you'll remember what T cells and B cells, white blood cells, that's about it. But in terms of these Golgi things, just forget it. I just want to show you where things are. And then so I can give you a three-dimensional architectures of how all these things work together. This is a common cell in our body. There's one part, it's called a nucleus. And there's a firewall around the nucleus. Outside of it, it's a cytoplasm, and this is the cell wall. And this is the cartoon diagram of it. The nucleus is here, and the firewall, it's here. I don't know whether you see the red dotted line. So that's the demarcation between the nucleus and the cytoplasm. The nucleus is where we have our DNA, and the DNA, this is how everything works if you want to make a protein. The DNA will make an instructions called messenger RNA, short for mRNA. And then the messenger RNA will be allowed to get outside of the nucleus into the cytoplasm. And then there's a factory that allowed us to make protein. You see all these tiny little dots over here? These are the ribosome. They are peptide factory that allows you to use the template of the messenger RNA, and the messenger RNA will latch on to this factory of ribosome, and then the amino acid will just link up one after the other based on the message or the template built into this messenger RNA. So messenger RNA is the little string, and then you'll find beads of amino acid that allow the proteins to assemble. And once these proteins get out, and it will then go into a packaging factory, which are these Golgi apparatus. And through this packaging factory, then things occur. So in this particular example, we're making a receptor. The receptor is called ACE2 receptor. It is a receptor how we regulate, the blood pressures and all that. But this is the same. This is the same portal where the virus is coming into our cells. So here is a SARS-CoV-2 getting close to the receptors, and then it get into the cells through a little bubble, and the bubble has acid in it, and before you know it, the messenger RNA of the virus, now not the messenger RNA of our normal protein, but this is the viral messenger RNA, get into it, and it hijacked the same peptide factory and start making protein for the virus. It doesn't benefit us, except benefits the virus. It's making these proteins and then go through the same packaging factory, and before you know it, you got the virus budding out of your cells. This particular cells is a lung cells. So this is a top-down diagram telling you that the virus coming in through the ACE2 receptor, release the messenger RNA, hijack the protein factory, and then making virus. And then that's how you get sick. Now, it doesn't get into the nucleus, right? This virus, it has nothing in it to allow it to get into the nucleus. There are other viruses can do the job. And so the first questions that I always ask is, oh, before I do that, I want to let you see this. Can you hear the volume?
No.
No volume.
No volume.
Hang on 1 second, I don't know. I'll be the narrator. You actually see these messenger RNA inside the virus. Seems like there's a audio issue. Okay. Give me 1 second. All right, never mind. So I'll narrate it. So the genetic materials are inside. There's a protein shell. And then there's an envelope. On top of the envelope are these viral spikes. So there's a droplets with the viral particles and getting through your nose, go through your nose into your lungs. And these are the hairs in your throat, and they get in and latch onto that ACE receptors that I just showed you. And then it gets through the membrane inside. Now you're inside the cell in the cytoplasm. Unlike the flu viruses, it can get through into the nucleus. The coronavirus cannot get through to the nucleus. So it stays outside and then hijack the ribosome to start making proteins for himself, for itself. So these are the proteins it's making. And it's making all the necessary ingredients now. And all these are the Golgi apparatus. And then all the ingredients for the virus are now available underneath the cell membrane. And then it starts budding out and then form a functional virion. And that's how the virus are formed. And that's the particles you see. So the first questions we have is, since the SARS-CoV is an RNA virus, and some RNA virus that people heard, like HIV, is the virus that's causing AIDS. It is also an RNA virus. This particular virus, HIV, do integrate with the viral DNA of our cells. So the question is, if you get a natural infections, will COVID-19 change your DNA? The answer is no, because it doesn't carry anything in the virus to allow the virus to get into the nucleus and mess around with your nucleus, the DNA in your nucleus. So rest assured, that's not going to happen. In fact, HIV retrovirus is the only virus I know that can do that. No other virus is doing that. That's why it's so dangerous. That's why once it get the HIV virus get into your DNA, it integrate with it. It can stay there for many years. And people who have AIDS, not only they have AIDS, but they also have a lot of malignancies, lymphomas, Kaposi sarcomas. There are a lot of cancers related to HIV infection. So this is again another view of the virus. Now I'm showing you the coil. This is the RNA coil inside the virus. It really doesn't, it's a minimalist because it doesn't carry anything else that will make it into a intact virus. It lacked the raw material, the protein factory, and the packaging factory. So all it does is hijack our cells to do all the work for them. And so that the virus using his own messenger RNA direct our protein machines to make all the protein necessary to make a viral particle. So it is a hijacker for that matter. It is also a lock hacker. All it does is mutate or change the lock every day, mutating to change the lock, so that it can be lucky that it opened doors. The doors that open in this case is the ACE receptors on the cells of our lung. And for flu virus is the sialic acid. The SARS-CoV, you probably heard in the news, they don't mutate very fast. Twice a month. I forgot the name. That maybe twice a week or twice a month. It's very, very, very slow. Flu, it's a lot faster. HIV that's causing AIDS is really fast. Now, all these viruses know how to change every day to see whether it's lucky enough to open up your lock so that it can get into your cells. I'll give you an example. This is a Ebola virus, and the Ebola virus normally in the proteins it will carry a particular amino acid in the chain. So the wild type will carry this. The variants know how to change one of the protein in this case, and make a variations of the proteins. The folding is a little bit different. Because of that one change, it allows them to gain access to the human cells more effectively. In this case, a cell is called dendritic cells. It's right here. Because of this gain in efficiency, Ebola virus in 2014 has a shift in the variant that caused it to have an outbreak in western parts of Africa within a very short period of time. Now you would think that, oh, mutations, that cannot be happening every day. No, this is happening with COVID-19. virus. And I'll give you the example next. So this is yet another view of, I don't know, a lot of artists want to do different things. So this is, instead of a coil, I'm showing you the RNA like this. Part of the spike is controlled by the pink area. And then in addition to the spike, there are also other elements on the viruses that are also important. Now, the whole world is focusing on the spike because this is the areas that will latch onto the ACE receptors to gain access into the human cells. So that's why we're focusing all of our efforts into making vaccine now, just tackling this part so that the virus cannot get into our cells. But the other proteins of this virus is equally important. That's what I call the broad spectrum immunity, is that when we get a natural infections, we have a broad spectrum protections against all of these elements in addition to spike. We are making a vaccines now only for the spike and nothing else. So this is why I call it a narrow spectrum. It has nothing to do whether the vaccine is strong or weak. We have very strong vaccine, but we have a very narrow focus. All right, so that's I just want to make that distinction. I hope the audios come back. It doesn't. So this is a spike protein. You actually see that the spike proteins like flowers, right? So this is the part that will attach to the ACE receptor. And it has three parts. It's like a robot. It looks like an alien now because it's going to open up and close. When it's open up, it's more accessible to the receptors. So Ruben is he's saying that it's like a flower. When it's open up, it's a lot easier to latch onto the receptors on the cells. And when it's open up, in this view, it can then attach to the cells and start the infectious process. So last year, in the beginning of the year, there's one change. It's called D614, and that's the story I want to tell you about. So 614 is one of the amino acids on this spike. And if you take the spike proteins and if you unravel this spaghetti, you actually result in a necklace, a broken necklace. And this necklace are studded with these amino acids. And these amino acids, nothing but, you know, this is one amino acid, another amino acid. They are a bunch of carbons, oxygens, and hydrogen daisy chained together. So a stretch of 15s, it's called a peptide because they're small. And I want you to remember that because at the end of this talk, I want to show you this is really important. This is really the heart. A peptide is really, or 15 amino acid stretch of peptide is very important. So for this particular story with D614, it's in position 614 of this 1,273 amino acid. One of the amino acids represent an aspartic acid. Forget about what this is, right? It's just a bunch of ball arranged in three-dimensional way. It's aspartic acid. I don't know why they abbreviated D as aspartic acid. It position 614, and that's in January when the virus first came out of Wuhan. Within 6 months, one of the variant decided changing it to a glycine. I'm having a ball because I can all of a sudden gain access to human cells better. So the D614 represent the changing from aspartic acid to a glycine. And with that, the flowers open up a little more. You see this in the D614, there's nothing over here. With G614, now one part of the flower petal is sticking up With that, it gained faster access to our cells because it get into the cells faster. It can make more viruses. So when this guy coughed, it coughed more viral particles and become more infectious. So in January, it came out of Wuhan. When you sample every viral sequence, you always see this D614. And gradually, when it gets to May, if you sample the viruses from the patient, it all changed to G614. And with that, we now have a virus that can spread faster and more virulent. And this is the virus that we have today. And this virus is now changing, because now you hear the UK. This is not the UK yet. The UK is coming. So let me do some housekeeping things. You hear a lot of people talk about R naught. R naught is the reproductive number of a virus. It tells you how you compare one virus to the other. If the virus have a high number like measles and mumps, 18 or 10, it means that from one person, it can spread to 18 persons easily. SARS and COVID, it's about 4 now. When it came out, it wasn't 4. It became 4 slowly. So this change from D to G bump up the R naught value by about 1. Right? It's hard to calculate, but that's what people think. Okay. So what I just told you is a kind of mutation called substitutions. When one amino acid gets substituted with the other or replaced by the other, it's called substitutions. And you're going to hear another substitution, N501Y. So position 501 amino acid changing from asparaginins to now tyrosines. And this is the core of the UK and the South African story. So this is N501Y as a mutations right at one of the top of the spike. And this is the receptor binding domain that you heard about, RBD. And there's another kind of mutations on the side of the crown that what he's saying that instead of adding something changing, it just disappeared. It make that part of the protein disappear. We call it deletion, right? So the virus be able to delete certain parts of the surface structures, like here. It used to be here when after the infections, I may have antibody to recognize this part of the structure, and all of a sudden the virus said, well, you're not going to do that anymore because the virus, which have this kind of structure, the antibodies latch onto it, all those viruses are gone. The remainings are the mutants that carries no proteins in this part of the spike structure. And so guess what? Even if you have antibody after infections, if you get infected with this virus, you're going to get reinfected again. This is what we call immune escape. In this particular viral construct is deleting a position 69 and 70. So it's missing now, instead of changing the amino acid, it's now missing 2 amino acids on the surface. And this has the name of mink cluster, and it's an interesting story. So again, in Q2 last year, it make a change to make it itself more virulent. Virulence means it's either, it kill more, it spread more, it be able to make more viral particles, just making the virus more angry, right? That's what it means, virulent. And this is the UK variant. I really don't want to talk about the geographic location. That's, I encourage everybody to talk about B117s and 501, and so on and so forth. And this is the story of the mink. I don't know whether you pay attention to the news last year. In November timeframes, the Netherlands start finding out that in the mink farm, there are human to mink passage of the coronavirus, and there are mink to mink passage, and the mink then also passing the variants back to human. And there are 214 cases of human transmissions, and that gets the government very upset. They decided to call 17 millions of the infected minks. And this particular mink has a profiles on the spike proteins that has the deletions mutation in addition to 3 substitution mutation. So there are multiple changes going on. This is what you get if you allow mutations to go around in 17 million infected animals and pass it back and forth. This is what intense farming would do to you. So this is the article just came out, I think, last month. And the study of this particular UK variant is now very worrisome because it increased the death per 1,000 cases from 2.5 to 4.1. And it also, that translates into a 32% to 104% increase in risk of death. So this is so when you see that the deletion 69 to 70, and this is part of the N501V1, and this is the one that we are now gaining ground in the United States. With that, you're now bumping up the R0 value to about 3 to 5. So now, instead of infecting 2 or 3 people, now you're infecting, more. So the virus is not only have immune escape, but also now gaining function. So this is where we are today. We have 3 different variants. So actually 2, because the D614 is built into the B117 and B1351. And you hear these all the time. We're now changing terminology to something different. It's called now VOC, it's variant of concern. And so you hear that in the news all the time now. If it's a 01, it's the 501, V1, it's the 117. If it is V2, it's the and so on and so forth. And this is the South African, this is the UK variant. And here you see that the study just came out and I just show you it gained in the killing power, and the transmissibility also increased. So is there ground to be scared? Well, this is not a very, in terms of fatality rate, it is not a high killing virus, 1.4 to 3.8% in case fatality rate. Compared to the sister virus, which is the SARS-CoV, they don't call it 1 because it's implied. Without 1, it's the first one. This is the one that came out in 2001. It has a fatality rate in the clinics about 9.5. And the MERS from the camel, it's really bad. It killed 1 in 3. So I want to introduce you instead of R0, the R0 will have, when it's greater than 1, it allows you to compare from viruses to viruses, which one it's bad. Measles and mumps worse than SARS-CoV and worse than hepatitis. So I told you that it gained the R0 number. But this number can only work when there's no, assuming that we have no intervention, we have no antibody, we have no vaccines, we're not doing social distancing. So in the real society, we don't talk about R0, we talk about RT. It's the effective reproductive rate of the virus in the view that we're covering our faces, we have social distancing, we have now people naturally infected, like my niece last week. She's now one of the N, has natural immunity. Now there are a lot of people getting vaccinated. So we are now building a firewall around the viruses. The heavier the firewall, the slower the virus is going to spread. And this is exactly what we need with the vaccines, to cut off the virus so that they're no longer spreading like it was in last year. And by not allowing them to commingle, you're also not allowing them to mutate too fast. So before I concluded the viral story, I want to show you this. This is not the entire list of RNA viruses. These viruses only have RNA, have no DNA. Coronavirus is one of them. This list of virus are all bad, all right? And the one that I highlighted with red is on the WHO pandemic list. So, you know, I don't even know what this virus does. I heard about it when I was in school. I heard about this, but there's no information. Now it seems like it causes outbreak once in a while. And I guess Ebola is here. Marburg. And you hear about dengue, hear about Zika virus, chikungunya, and influenza. Influenza, it's bad. Now, all of these viruses are what we call zoonotic, because they either go into an intermediate we call vector, and either the bat is the reservoir or the camel is the reservoir in case of MERS. In SARS-CoV, it's the pangolins, the ferrets, hamsters, civet cats, and we know mink for sure. A lot of them go through bats, some of them go through mosquitoes, some of them go directly hitting mammals. And in the case of HIV, even though the initial years it go through primates, and now they don't need the primates anymore, the HIV virus infect human, no different than the SARS-CoV, right? So there is a link to these intermediates. So if we get close to them, we're asking for trouble, right? So let me stop here, and I'm going to start talking to you about vaccines, and then come back later to talk about this story. So here it's where things link. We now see that the bat, the mink, and the intense farming causing some troubles, moving the virus from this end to this end. We're also now seeing with either natural immunity. Now, I credited this to natural immunity for moving the G614 or the N501, because the vaccine hasn't really created that much pressure yet for the virus to change. It is the natural immunity from the folks who have recovered from the virus last year. It's causing the virus to think of ways to escape the antibody that are now circulating in the community. And they're very good in finding a way to escape. So what about the vaccines we have today? So I'm going to tell you how many are there, how they work. There's a common theme to these vaccines. They only do one thing. Some do better than the other. And then tell you the current available vaccine in the US, how they compare, what are the risks, and what's the reward. So if you go to the vaccines tracker, you will find that, wow, there's not only 3 vaccines. There are actually 12 vaccines approved around. In fact, I think now we're on 13. This was last week. And then you have another 22 in phase 3 ready, it's coming up. And then there are another 60 some vaccines in the work. And your question is, why that many? Because we want to have broad spectrum vaccines, and the vaccines we have, it's only work on the spike. And these are the 12 approved vaccines, and here are the familiar one from Pfizer, Moderna, Johnson & Johnson's, AstraZeneca, and the check marks are the vaccines that I want to talk to you about today. And so these are the 22 that are in the phase 3 clinical trials and up and coming. So I want to talk to you about 4 classes of vaccines. And the old ways of making vaccine is you're taking a virus, you knock them dead, right? You fix them with formaldehyde chemical, and the virus are intact particles, but they are dead. But before you have to kill them, you have to grow them. So we grow them in eggs. You saw this, 10, 15 years ago, we couldn't find enough eggs to grow viruses. You inoculate the eggs, and then you harvest the eggs, and then you chemically purified it. And that's how we make vaccines for 60 years. So we know this world very well, but they are not very good. In fact, the Sinovac, it's one of the prototype. It only have 50% efficiency. So it's not very good. We know they're not very good, right? And another way to make a vaccine is you strip off the proteins from the virus, and then you bundle them in a small ball, and then you give it to the patient. And these are called subunit protein vaccines. We have 30 years of experience making this kind of vaccine. But Novavax is another one up and coming. I think they're going to get approved in about a couple of months. They have a new twist to it, and I want to tell you about it because it's up and coming. You heard about the messenger RNA vaccines. It's nothing but a messenger RNA enclosed with a lipid core, ball, and they are Moderna and Pfizer. In the R&D community, we've been working on this for 15 years. So we know a lot about the science, but in terms of products, we have less than 1 year experience. This is really the new platform, the new kids on the block. Now I want to show you these diagrams. These vaccines, they don't need to go into the nucleus. They just go into the cytoplasm, do their things, and then that's it. All right, so never, don't even have to ask the questions, are you going to mess around with my DNA? None. This class of virus, it's called viral vector vaccines. These are the Janssen, the J&J, the AstraZenecas, the Russians, Sputnik, the Chinese, CanSino. They do go into the nucleus, but they don't integrate with your DNA, and they turn around, finish their work, and come back to the cytoplasm. They also do not integrate with your DNA, and they're making sure that it doesn't happen. I'll tell you why. They're using this, another virus, as a Trojan horse. You heard about that in the news, and I'll show you how that works. We have 25 years of experience working with this particular viral vector, right? But in terms of product, we only have less than 5 years of experience. Janssen has a Ebola virus that was approved maybe a couple of years ago. So we have some experience in real life how this kind of virus, this kind of vaccine works in the real world, but it's relatively very new. And then the other kind of DNA virus is also may not be up and coming, but I'm sure it'll be up and coming in about a year. And you hear about this. There's a San Diego company. It's called naked DNA. So instead of putting the DNA inside a vector, they just give you the DNA. Simpler that way. We have many years of experience, but we have never made a product. So this is really new in terms of product, right? So I'm going to go through each of them, and I think you appreciated how these vaccines are made. And I'm going to tell you how these 3 vaccines, how they are made in the real world. So now if you have a, if you are a beer lover, if you have a fermenter in your garage, you can also make vaccines using the fermenter. All right, not that I want you to do that, but essentially the equipments are very simple. This is the virus with a coil. If I take the coil out, this is the messenger RNA. On the messenger RNA, there's a stretch of the RNA that code for the spike protein. So if I take this little stretch out, then now I have isolated messenger RNA. If I find a way to express this into the spike, I get the protein, right? Just that simple. So if I do a genetic modify techniques and I engineer this single strand into, and I just reverse engineer from a RNA and make it into a DNA. This is done in the lab every day. There's no secret to it. It's very routine. Once you make the double-stranded DNA, you can then marry with the adenovirus, in this case, the Trojan horse. Right? So this is the DNA vector vaccines, the Janssen, the AstraZeneca's, the Sputnik Russian vaccine. So this part of the adenovirus as a Trojan horse has one part of the adenovirus genomes removed. So this virus is now a weakened virus. So I would call it a defective virus. Once it can only get into our body, but it cannot make baby on its own. It just cannot. It's called non-replicating viral vector. Now I want to make you aware of these things because among the 68 other vaccines that are coming, some of those vaccines use replicating virus, which means those Trojan horses can likely make more Trojan horses, and it's there are a lot of cute ways to skin the cat. But in this case, these are non-replicating virus, and then you put it into the adenovirus, and you put it in your fermenter, and you need a surrogate human cells to grow this. And I'm going to tell you a story. There are some noises around this from the churches and the pro-life people, and then you make a lot of little adenoviruses that carry the spike DNA in them. There's no protein, but they carry the DNA for the spike proteins, and then you vial it, you inject it into muscle. The muscle cells start producing these spike protein, not the viruses, but the spike proteins. And then our immune system will look at it and say, wow, you're not supposed to be there. I better make some immune response on it. So messenger RNA vaccines. Well, here I'm already have the message. Simpler, I just wrap it in a lipid surface, and then I use chemical methods to make more of these, put in the vial. It's really a lot simpler. There's no growing of anything, really simple. And then you inject same way, and you make spike proteins, and then your body responds. Now, there's no reason why the vaccines get into your body won't directly stimulate the immune cells. It could, all right. So I'm just, for the most part, if the vaccine drain into the lymph node, it will directly engage the immune cells. But I just show you, regardless, you are expressing these spike proteins. So the cute part about the Novovax vaccines, which is a subunit vaccine, so they don't shoot any DNA or RNA into your arms. They only shoot the spike protein. The way they do it is they also make DNA, and then they put it in another Trojan horse called baculovirus. The baculovirus, they don't infect human. They only infect insect cells. And this particular cells, SF9, came out of moth. So, scientists are very crafty. So, they use these cells that carry the spike DNA, and then they make a lot of it. They produce a lot of proteins, and then they make it into a flower arrangement. And this is essentially, if you go to the websites of Novovax, they show it this exact arrangement. So there are a lot more units of these spike proteins that are presented to your muscle cells, and you stimulate immune response this way. All right. So this is how they make. So in the following sections, I want to tell you one by one how do these. So you inject it into your muscle, and it get into your cells. So where do they go inside your cells? I want to show you that. And what is the formulations of these vaccines? Are there any allergens in there? We hear people getting an anaphylaxis. What's going on? What's in there that may potentially precipitate all those adverse events? So let's get some ideas. I'm not saying that I know it all, but I can only tell you some of these things that are already in the journal publications. So what issues are there? So this is, again, the viral DNA viral vector vaccines from these companies. Once they get in, they have to go into the nucleus, right? In the nucleus, they will make messenger RNA, and then the messenger RNA will come out and then make proteins through your ribosome in these tiny little dots over here, and then package it through the Golgi into proteins. And these peptides, these proteins, are now expressed on the surface of the cells. And your immune cells look at it and say, oh, wait a minute, yesterday I didn't see these things on you. Today I'm seeing a lot of these. It must not be normal. I must do something about it. And what doing something about it is what we call immune response. Right? So these are the companies making the DNA vaccines, and they all carry the AD in it because of adenovirus. So the payload of this inside the DNA of this adenovirus are just DNA, the DNA that code for the spike protein and nothing else. The Trojan horse is the adenovirus. And for Janssen, the adenovirus is coming from human. For AstraZeneca, it's coming from chimpanzee. And this is another version of adenovirus 5, is the older version of adenovirus. And then the Russians are smart. They just combine the patent from Janssen's and the Chinese patent and make their own. And so in the first injection, they give you the adenovirus 26 that carrying the spike. And the second injection is adenovirus 5 carrying the spike. And that's how they do things. I want to tell you this because there is an issue with these adenovirus, because these are common flu or common cold virus. You and I probably have been exposed to adenovirus off and on throughout our life. So a lot of us do have immunity against this adenovirus. So what happens if I have antibody towards the adenovirus? Well, one possibility is before the Trojan horse get a chance to dump the payload, my antibody already attacked the Trojan horse, and the Trojan horse will not be useful. All right. So, it may not be the best vehicle to do these, but this platform has been used now in many gene therapy. You're going to hear about it for a long time, because a lot of the cancer therapy now coming out using the adenovirus as a construct, and we have 25 years of experience with this thing. All right, so I don't want to poo-poo it and say, this is no good. A lot of goods will come out of it, but there are issues. Just like every solutions will come up with other problems. So what is the formulations of these? And if you look, go into the FDA meetings with these companies, they will show you the formulations in a very simplistic term, and I'll show you one soon. So in this, you can see that there's nothing really in there that will cause issue. A lot of salt, a lot of proteins. You know, a lot of these are used in many drug formulations. You can't really pinpoint that it is an issue. But polysorbate is not an issue, but the parent compound of polysorbate 80 is a problem, and that is the story for messenger RNA vaccine. But polysorbate itself is not an issue, so I really don't see anything that will cause anaphylactic shocks or any allergic reaction. In fact, I'm going to show you data. Janssen's product probably have the lowest adverse reactions ever in all the vaccines we have on the market. And I'll show you that data soon. The only thing that is really different is all of these are having the human adenovirus, and only AstraZeneca have the chimpanzee virus, and don't know why. They have problem the last 2 weeks. They're in the news now. I think there were 7 countries stopped vaccinating people with the AstraZeneca vaccines. And I think today, all of them are back on track. Seems like the blood clot, they are not anaphylactic shocks nor allergy. This has to do with some vaccine recipients, they have blood clot. But sounds like the number of the blood clot individuals are not high enough. And they have now reevaluated and say, you know, there's no difference between the folks without the vaccines and with vaccines in terms of incidence of blood clot. And now they are back on track. So the questions here is, are there any allergens? I just told you there's nothing in there. So the formulation is quite clean. Okay. So let's continue with the DNA virus. And so, you know, just give you, they're non-replicating, they're common cold virus, the adenovirus has been used in Ebola vaccines, so, and has been FDA approved. Now, all the vaccines we have today they're not FDA approved. They are approved for emergency use only. They're not approved yet. So there are still phase 3 studies ongoing, and they will have to be finally approved by FDA some months or year later. But we only approve them for emergency use. But Ebola vaccines using the adenoviral constructs has been approved for FDA. So we do have real life experience with this construct. And Merck and J&J both have it. And I don't know whether you know it, Merck is actually now making Johnson & Johnson's COVID vaccines just to get things moving. I'm going to skip this because the audio is not working. So here's the issue number 1 I told you about, because a lot of us do have adenovirus exposure. So we have immunity against the adenovirus. And if we have it, then the antibody to adenovirus may destroy the Trojan horse before the payload can be delivered. So that's why J&J use a AD26, because it is a rare subtype. So not as many people are exposed to it. Most of you are exposed to this are the South African folks. The rest of the world have not been exposed to adenovirus 26. That's why AstraZeneca find the adenovirus from chimpanzee, because we're not exposing to the chimpanzee adenovirus. So they are safe. And the Sputniks, they think if I have problem with one, maybe I won't have problem with the other. So they use a combination of 2. But even this may not be an issue now. If we use this vaccine over and over again, then all the vaccines recipients will sooner or later develop an immunity to the adenovirus and will render the vaccines, you know, I won't say useless, but at least it'll be compromised. The second issue is with the DNA vector virus has to do with, remember I told you the surrogate has to go into human cells and then go into the fermenter. The surrogate human cells are fetal cells that are collected from 2 aborted fetuses in the 60s. Sorry, I can't get you Ofit's audios explained a little bit better. These are from 60s, and the scientific communities have been using these cell lines, these 2 cell lines, over and over again. So there are no new abortions done in recent years to support this kind of product. These cell lines were collected from way back in 60s with proper informed consent. And so AstraZeneca is using a HEK 293 cell lines. It's a fetal cell lines from kidney. And then the J&J using the PER-C6, and this is a retinal eye cell lines. So they've been in use for 40, 50 years. So this is the only news I found. People say, oh, the Vatican's against it. I don't know the current positions, but in the Vatican news, all I get is seems like they approve of it. And in the audios, they did say that it was approved. But there's a lot of noise. But the question is, if you're using these cells in the manufacturing, will then the finished product carry a remnants of these cells in the vaccine? The answer is yes. There's a trace amount of these human fetal cell line proteins, or DNA, not protein, likely fragments of broken nucleotides, DNA material in the vaccine. And so that's, you're gonna have to live with that. But whatever the contaminant is well within the FDA guidelines. One of the notes is the messenger RNA vaccines do not use any of these cell lines. So it's, so you don't have to worry about that. So while we're at that, it's a good segue to talk about the messenger RNA vaccines. How do they get through the cells? What are the formulations? Are there any issues? So this is the DNA viral vector I just told you about. So now let's look at the messenger RNA. It comes into the cells after vaccinations, and then it will shed the outer shell, and the messenger RNA will then, it doesn't have to go into the nucleus. It just skirt around, and then we'll find the ribosome, these little nodules over here, and then start making the proteins, then package the proteins in the Golgi and start expressing it on the surface of the cells, and it end up the same way. Our immune system see these are foreign proteins and start reacting to it. This is Novavax, even better, because I told you that they don't even have DNA or RNA. It's just a bunch of a protein ball. Once you inject it, it goes directly to the immune cells and start working on the immune cells. So these are the 2 messenger RNA, and there are more messenger RNA vaccines coming in those 68 on the trial. I really want you to hear directly from Moderna. Okay, I'm not getting, but I'll narrate it. There are some features here I want you to.
So Herb, to get the audio, you probably have to turn on Soundflower. I'm not sure if you're familiar with how to do it.
Because I tested it beforehand. Seems like the audio is okay. I don't know why.
Well, going through Zoom is a challenge on video audio, and you typically have to change the audio source to Soundflower.
All right, but let me just narrate. So this is the messenger RNA that corresponds to expressions of the spike protein. And then they make these, cover this with a lipid coil. It's called lipid nanoparticles, and under the electron microgram, they're very uniform, right? And then they make it in these large, they're not fermenter, these are now chemical tank, because it's just the lipid enclosure with the messenger RNA in it. So it's very simple. They claim that the formulation is nothing but just water, sugar, and some FDA-approved buffer. But of course, there are multiple material in it. I'll show you what they are. And they have no preservative. That's good, because some of the mercury preservatives that use in the older vaccines are not here. So when the vaccine comes here, it stimulates this cell. These are immune cells. It's called antigen-presenting cells. And then once it gets in there, it will make proteins, and then the protein will show up on the periphery, like here. And I show you this, in previous slides. And then once they express, they can then release into the surrounding. Now, remember these three diagrams. I'm going to show you at the end. This has to do with the handshake between these molecules, but let's not worry about it now. It has to do with how we engage the immune cells to do the final work. So you're going to hear from, if you were to listen to audio, you'll hear the T cell, B cells. Every companies tell you the same story. And that's it. I'm going to stop here because there's no point. So these two vaccines, this is the formulations. So you heard that it is very simple. There's nothing in here that will cause allergic reaction that we see, except for polyethylene glycol. These are polymers that are used to increase the life of the drugs as it go into our circulation, because you want the drug to stay in your circulation long enough to do the work. So if they disappear, it's called a half-life. If the half-life is too short, then it's not doing the work. So polyethylene glycol has been used for eons in the drug industry for that purpose. It's a good bulking agent. It's very inert, except some people are allergic to high molecular weight stuff, right? Now, I'm not saying that this is the culprit, but it has been, I think the article is underneath at the bottom, and they've been reported. And in fact, the Allergy Society put out an article some years ago and talk about, the polymers does have issues with certain people, but only with the high molecular weight. The low molecular weight is not an issue, right? So the question is it the allergy that we see, because of the polyethylene glycol? There's some suspicious, but I cannot tell you. But this stuff is everywhere in household products as well as in pharmaceutical products. So just to sum it up, 4 different types of vaccines. These 2 don't need to go into nucleus. These 2 do have to get into the nucleus and come back out to the cytoplasm, but none of them mess around with our DNA. These 2 require Trojan horses, and these 2 don't. So a lot of questions in the community is, well, can I get COVID-19 infections, by receiving the vaccines? No, you cannot. There's no viruses. Well, there's virus. The only place a virus in here, but they are dead virus, and we've done this for 60 years. We know how to kill viruses. This only carried the proteins and no DNA, RNA to change anything. This has the messenger RNA for the spike and no DNA, no viruses. This same thing. No, there's no viruses. The only virus is in here, but it's an adenovirus, and this adenovirus is non-replicating. It is a one-way suicide missions. If I have to characterize this Trojan horse, it only get into your cells once, then it get degraded after it dumped the payload, and that's it. It will not make more viral particles. So no, you will not get COVID infections by accepting vaccine. So then it brings us to the efficacy. I'm not going to show you efficacy beyond this slide because you hear that from news, every day. The mRNA, 94, 95% effectiveness. The DNA, for Janssen, it's the lowest among, and it's only 66%. And I do want to have you do some critical thinking to see that, is this fair comparisons, 66% versus 95%? Doesn't matter, right? This is the Russian Sputnik, and a lot of people poo-poo about it, but seems like from the publications, the 2 injections of 2 different adenovirus give you 92% effectiveness. So it's not bad, except I haven't seen peer review articles on it. So I'm still waiting to see, from scientific validations how to perform. The Novavax, the flower protein, it has 98, but there's no official words coming, but this is going to go through FDA approval cycle soon. The AstraZeneca, as you heard, it's 70%, but it's weird. If you give 2 full dose, you get 70%. But if you get 1 half dose and 1 full dose, you get 90%. So there's some theory behind it. People are guessing, but it tells you that even though with Janssen's 66%, if you give, and it's you only giving 1 dose, if you give another a boost, this could easily be the same story. Right? You see the prime and the boost, you get 90%. Easily with the prime and the boost, with the Janssen, you'll also get 90%. It doesn't surprise me. The Chinese inactivated virus only get about 50% mark. So this is not really good, because if you vaccinated with a virus this poor, guess what happened? Think back to the diagram I show you. You'll only be able to weakly control the virus that the antibody or some of the weak antibody can bind. And then the variants will probably escape. You may cause more problem than good with a vaccine. So I want to get to the questions that people ask. Well, because this is real, right? I remember the day when Johnson & Johnson came out with the 66% effectiveness. A lot of people already signed up to do clinical trials with Johnson & Johnson, and they quit because they said, why am I getting your vaccine where the effectiveness is only 66%? Why don't I just, get the real vaccine? So this kind of comparisons, it's very superficial, but the question is it fair? So I want to give you the rundown on what I think. So if you look at the results coming from Pfizer and Moderna on the messenger RNA that give them the 94, 95%, they really don't split up the patient populations by mild, moderate, and severe. They just lump everything together and say, well, it protects you from getting COVID 94%, 95% of the time, right? Regardless whether you're mild, moderate, and severe. They also, at the time they were doing the studies, it was in the time frame of from June to November timeframe before they closed the studies. At the time, we only have the D614 variants in the works circulating in the world. So these vaccines only tell the stories of how it affects the D614, not so much about how it affects the nastier variant. So question is, well, what is your efficiency against the variant? That's the question. So I'll get to that. For Johnson & Johnson, they came out, they said, well, I don't care about the mild disease. I only focus on the moderate and severe disease. And they got the 66.3%, after 14 days after one injection. At the time they were doing the studies, the UK and the South African versions were going rampant. And they find out that vaccines In the US, it's about 74% effectiveness, but in areas where the B135 was rampant, it's only have 52%, right? But they're telling you much better coverage than the Pfizer and Moderna. They said 100% effectiveness preventing hospitalization, 85% effectiveness in preventing severe and critical diseases across all regions. And more importantly, once you get vaccinated, you're not going to die, regardless, right? I mean, these are important statements to make where the study with Pfizer and Moderna never get there. Of course, they're now doing studies to catch up. So how does the Moderna and Pfizer stack up with B117 and B135.1? There's some coverage on 117, but when it comes to the 351, it's not so good. It's about 50%. No, not any better than J&J. But of course, now they are working on a booster shot with the B1 351s in the mix. So they're gonna get better in coverage. And of course, Johnson & Johnson also doing the booster shot. Now, there are studies they're doing now. Instead of one single shot, they're giving you 2 shots. And we see that with Sputnik, with 2 DNA vaccine shots, you can bump from 66 to 90%. It doesn't surprise me. So is it fair? So in order to be fair, we need to have an apple-to-apple comparison. Now, even with this study, it's not apple-to-apple, but this study shows you something different. This was done in 2 nursing facilities in Connecticut where they have outbreak of COVID last November or December. And these patients have a lot of complexities in the disease, from cancer patients to just a long list of things they have among these 463 patients. The study came out that for those who receive partial vaccines, so which means they're not getting 2 doses of the Moderna or of the Pfizer, but they get 1. It turned out those who get partial vaccinations still have less infections than those who never get vaccinated. So these are the non-vaccinated. There are a lot of infections, but the vaccinated group seems like the incidence of COVID infections are much less. This is in facility A, facility B. So this tells you that even with a partial vaccination, you are somewhat protected, right? And so, statistician calculated 63% with a partial dosing at about the same time. So, if you look at it, you really cannot say that one is better than the other. I would say that they're all equally effective, right? That's what I would say. And in terms of death, there's no one died, even though they're very sick, nobody died. Side effect, yes. What do you expect? These are very sick nursing home patients. So here it is, partial vaccinations, you got 63%. Now there's another real world experience we have with the Pfizer, and this has to do with the Israeli data. So in January, they're pretty smart. They pre-arranged with Pfizer a long time ago, saying that when your experimental vaccine comes out, we want to be the guinea pig. Supplied us with enough dosage that I want to vaccinate the entire Israel. So they've started that exercise in January. At the time this study was done, they were up to 500,000. I think now they are up to like 4 million people have shots. They're the highest vaccines coverage in the world. And this is what they get. After 1 dosage, they get about 51% effectiveness at about 13 to 24 days. Minimal side effect, only 0.25% have complaints on side effect. And after the 2nd dose, it bumped it up to 94%. So this is the first real world experience we have with the Pfizer vaccines that it does work. It's a powerful vaccine. It does work to shut down or to at least prophylactically prevent viral infections, right? Now, there's some inkling that this also will prevent spread, but I think I would rather see more data before we jump to that conclusion. But it's good data here to show that not only prevent infection, but also shut down the viral transmission from one person to the other. And that would be the good news. And of course, here you see that the viral, the daily count of infections go down. Now, the world is all of a sudden have a downtrend, but then seems like in other countries there's reverse now in Italy, in some part of UK. So this is not the end of the story yet. I'll skip this. So now we're talking about the risks now. So what are the risks with these? You can go to the CDC sites and hang on one second. So these are the knowledge they give to the physicians, how to answer questions in terms of complexities, Bell's palsy and all that. Not very useful to me. There's another site you can go browse around and talk about allergy reactions. This is the site where the whole world in the United States, once they vaccinated, they encourage you to report adverse event. It's called the VASA system. It's a self-reporting system. So obviously some of the reports of the events in there have not been verified. So you're going to see a lot more reports in there than real. You get a vaccination, you say, I don't feel good. I didn't sleep well. I have a headache. You report it, but it doesn't matter to anything. But there are events in there. If you go in and look at it, it will scare you. All right, I have anaphylactic shock. I die. There are a lot of death, but I want to show you it's not. You really shouldn't go into the database and scare yourself. So I followed instructions and did a search. I just did a simple search. Tell me all the events associated with these vaccines. Janssen have 2, amazingly. I would say by now, I did it like a couple of days ago. I thought by now you have a lot more, right? Even though they're the latecomer. But Moderna and Pfizer, that's up to like 17,000 complaints about some events following vaccination. And if you look at it, some of them are not very pleasing, right? But if you look at the total events versus how many shots we've been giving now, we immunize like 110,000, all right, 112 millions, I'm sorry. So if Moderna have 45% of the shots, Pfizer 52, and Janssen's latecomer have 3%, you're giving 50 million doses here, 58 millions. Of course, 2 doses, some get 2 doses, some get 1. So, it doesn't matter. But with all these doses, you're getting 13,000 complaints. With 58 millions, you get 17K. Percentage is still very low. Now, if someone have significant adverse events, anaphylactic shock is significant. I don't care whether it's 1. You don't want to see that, all right? So I don't want to poo-poo it and say, that's not risk. It is risk. And there are a lot of complaints, right? So what's going on here? I want to show you, that's why you shouldn't go look at it, because CDC have investigators go look at these things every day, because what you don't have is based on the events that are logged in the database, some physicians will have to go investigate and qualify each case based on some criteria. Now, there are 2 criteria here. The first grading systems, it's about the severity of these adverse events. If it is grade 1 and 2, it'll go away in 2 days. Yeah, you have discomfort, but nothing to it. A lot of people after vaccinations have pain in the arm, and that's in this category. Some people have more severe events, and they lock in and they say, well, I have pain, I need Tylenol, right? So there's a little more involving here. I have a fever. Grade 4 are the ones who say, well, I really don't feel good. I'm about to have a sleep. I have headaches, I'm dizzy, I need to go to emergency room. So that's grade 4. Now, that is not enough. That just tells you that how serious is your problem. It doesn't tell you what problem it is. So then when the physician go in and query that database, they have to qualify, with the highest certainty. If I'm looking at anaphylactic shock, there's a Britain scale, and they would say, well, level 1 and 2, I'm sure there's something in there that I have to go look for it. If it is level 4, it's just a complaint, it resolves itself, don't worry about it. So I want to know how many, out of those 13,000 or 17,000 cases, how many are actually level 1 and 2, right? Now, granted that the 13 and 17,000 are all from grade 1 to grade 4. It's not just grade 3 and 4, it's just everybody. You after the vaccination, you sneeze, someone filed a report, and that's grade 1. Right? So this is a publication that just came out not too long ago, talking about people after vaccination, they have allergic reaction. I'm going to step up a little bit because of time constraint. So the CDC physicians went in there, look at all the reports, and they qualify based on this scale, and they find 100 out of the one, at the time they only have 1.9 million people got the first dose, and there were 157 out of all the complaints, 175 fit the descriptions of level 1 and 2. So something there that we have to investigate. Out of that, only 21 that are really serious and end up in the hospital, and all of that get resolved. One, I know it get resolved, but there's no information. Essentially, even with anaphylactic shocks, the serious allergy, you can get treated and nobody die, and that's important. But number of cases is not very high. It's 11 cases per million. So in terms of the safety, it's fairly safe, and especially they all get resolved. I'm not going to go through this calculation because I was going to tell you, based on what we have, just give you the bottom line. Essentially, by the calculation, the 17,000 ought to be really in hundreds because based on the experience here. But one thing that you get out of is after 3 million shots, you only get 2 complaints from the J&J vaccine. It's exceptional. I'm going to skip this. It just tell you, it's also low risk from another studies. I'm not going to go through it. So in summary, we have 3 vaccines. AstraZeneca is coming to FDA next month. I'm sure it will get approved based on what we see today. So by April, you can have 4 vaccines. Novavax coming out in 2 months. All of these have some efficacies, even with the worst variant, it's 50%. Now I want to give you this as a reference. Having vaccines in the 94, 95%, it's unheard of. Right? In the world of vaccine, it's a super vaccines. Most of the vaccines are not that great. If you look at flu vaccines, all these years, the most you get, it's about 60. I don't even see 70 here. All right, that's what you get in real life. Having the worst case against the variant at 50%, you're still doing some good, right? But for the most part, with these vaccines at the 90% range, it's really good, right? These are very strong vaccines. Now, I don't want to question, how broad, how narrow. Yeah, we're working on the spike because we don't have time to work on other stuff. But I hope some of the newer vaccines come out down the road will have a broad spectrum. We have 3 very high efficacious vaccines. What's your choice? Don't take it and risk the 2% death, or take it, even at the worst case, you're not going to die. But the side effect, it's a minimum. I don't want it. My recommendation is, yeah, take. It's my turn. I will do it. I won't even pick. So I'm going to give you the 3 videos that you can get from YouTube. The question here is, wow, you guys finished the vaccine in 1 year, when most of the time drugs, including vaccines, take 8 or 9 years. It must not be very safe because it develops so quickly. I've been in the business for a long time. Really, the whole world dropped the ball to do anything else, just focus on working on COVID. There are a lot of tools that you have to develop. And that's why it takes so long to do these studies, because not any lab can work on nasty viruses. It needs high securities to contain the viruses that you're working on. So most of the time, these nasty viruses only are worked on by qualified laboratory with high securities, right? In order for the whole world to work on these things, we have to make tools called pseudovirus. They work like the real virus, but they don't infect people, right? It gives you a surrogate endpoint so that you can now work instead of P4, you work at a P3 or P2 level security or safety, right? A lot of groups work on these tools to make these tools free and available. And that's why things happen so fast. Company dropped everything. You heard the news this week. The world hasn't screened for colon rectal cancer, breast cancer. We're not doing that. We're putting all the resources developing products only for COVID, all right? Recruiting patients usually take years because, people don't want to be the guinea pig, right? Even if you give them a couple $100, $1,000. So it's the longest time to get clinical trial. Phase 3 clinical trials takes 2-3 years because you cannot sign up patients. Here, you can just throw a rock at it, you're going to hit a COVID patient, all right? So it's easy to get. That's why things move so fast. The regulatory pathway, yes, they bend over backwards, but I want to show you this process. And that's why I want you to hear the videos, and I cue it to the point that you can listen to the efficacy, you can listen to the safety, because these are 8-hour tapes. This is the open meeting for the emergency approval meeting between the sponsor and FDA. So they start out with the company giving the data, and then the FDA, the independent studies on the data set and how they evaluate the data. And then the board go through the discussion with a whole bunch of vaccinologists. And they go through the process. There's no shortcut. They just do things much faster. All right. So I want you to hear that and get an appreciation that, nobody is just like, approve these things and haphazardly, right? I listen to all the tapes, boring. So the last section, I want to spend 10 minutes to tell you how it works. Actually, sorry, I'm going to be above my allocated limit. How it works. So this is the crux of the matter. So you have a COVID natural infections that get into your lung. It causes a lot of diseases, the lung, heart, the brain, gut, coagulation, you name it. The nasty, even though the fatality rate is low, it causes a lot of damage. It's contagious, it's uncontrolled, it's systemic. Once you have, I don't know when my niece is still here, and she just got through a course of this. So her body it's now making natural immunity. It's waking up the antigen presenting cells. These are the macrophages and dendritic cells. Just remember they are white blood cells and nothing more than that. So it's waking up these cells, educating them about the spike proteins. Yeah, there's something invading that you need to do something. all right? But this process, it's uncontrolled, systemic problem. What we should have done is introducing a sterile inflammation, nothing that will cause damage. It's controlled because it's local, only hurt your arm and only give you a swollen lymph node, for 2 days. But it's well controlled. It doesn't cause systemic diseases. And it's specific for COVID-19 because I'm giving you the spike protein of COVID-19. So this is the process the vaccine is doing for you, all right? The end result of this is all the same. It's beyond this point. It wake up an arm of the T cells called CD8 T cells, and these cells will go attack any cells that are infected with the virus. It will have a marker of these proteins on the infected cells, and the T cells will go in there. I see you. I'm just going to kill you. Now, every day, and my niece will tell you, because one day she'll have 103, 104 fever, and then the next day she has 102. The third day is less than 100, and the fourth day, finally, after remdesivir, her fever went away. Because when the virus come out, where do they go? They go to another cells. That's all they do. So now the virus come out. Some of them are killed by the antibody. Some go back into another healthy cells. And then the next day, another crop of viruses come out. You have fever again. And then it go on that cycles 2 or 3 days until the virus are all dead and you clear, right? That's the CD8 T cell cellular immunity arm. I'm just going to say it once, forget about it. It's just the T cell arm. The other arm is also T cell mediated, but it will wake up cells that make antibody. And these are the antibodies you hear about in the news every day. So, but it only tackle the viruses that are free flowing in your bloodstream. The viruses inside the cells, the antibodies cannot touch because it's inside the cells. So without the T cells attacking the infected cells, the antibody alone is not very good. So what's the vaccines of the goal? The goal is to reduce symptoms and stop transmission. I'm going to skip this, sorry. So am I telling you that all these vaccines doing the same thing, right? Oh, sorry. Right? They are doing the same thing to wake up the T cell arm, the 3B, and to wake up the B cell antibody 3C. So all I want you to remember is the path is 3A, 3B, and 3C. That's how you wake up the immune cells. So then why can't they be interchangeable? Well, the CDC recommendations do not mix these products now until we know the safety profile of these vaccines. And obviously there are ongoing trials to address the mixing of COVID-19 vaccines. For example, J&J has a phase 2-3 trials in South Africa to study the Adenovirus vaccines as a prime, and then follow with the Novovax protein vaccine as a boost. So you mix the two products, but that has to go through clinical studies. The Sputniks already did some kind of that. It's mixing the 26 with 5, and they get 92% efficiency, so that's very good, all right? Are side effects expected following vaccination? Well, you are creating a sterile, well-controlled, localized inflammation. Inflammation means you're going to have pain. Sometimes you have fever. Sometimes you have fatigue, but hopefully it's short-lived. All right. So yes, you're going to have pain. So just because you have pain, it doesn't mean that there's something weird with the vaccine. So remember in that diagram I show you, remember the handshake with the Moderna? All right. So I'm now going to tell you the story that is dear to my heart. It is the handshake between this white blood cells and how to talk to the other white blood cells to direct them to either kill the viral infected cells or to make antibody. And this handshake probably associated with 15 Nobel Prize stories. It's a very important part of the immunology it's also the brain behind the immunology story. And the handshake, all I want you to remember is it's managed by barcode. So here it is. So how does the immune system deal with constant threat of things like the variants? But that's not the question to ask. The question to ask is, how does the immune system tackle any kind of external threats? Any kind, right? Not only the external threat, but the internal anomaly that are coming up in your body every day. I want to impress you, there is a universe inside your body just as complex as the universe up there. External threats are not just the COVID, are any pathogens, viruses, fungi, parasites, bacteria, anything that we deem it's non-self, all right? The internal anomalies are even worse. Every day we have cells dying, but if the cells are not dying properly, it may become cancer cells. It happens all the time. But someone have to police that and say, well, you got cancer cell today, you're eating way too many hot dogs or barbecues, and the cells are not behaving, and I need to control your cancer cells before it becomes a problem. Immune memory is doing that. The proteins not folding properly, it causes Alzheimer's, Parkinson's, diabetes, autoimmune diseases. And then you have, unplanned injury, burn, whatever, it's causing tissue injuries. Someone have to say, I recognize you have a problem, but the way that we recognize there's a problem is to first recognize who are you. So our immune system have to memorize not only all of these things from external that are lumped under the umbrella of non-self, it has to remember who you are. In the first 20 years of our life, we almost have to remember every single tissues that are growing in our body. And not only that, in the last 10 years, we find out there are 50 to 100 trillions of friendly bacteria that live in our gut. You cannot kill them, right? But the immune system know who they are. And these are called the microbiome. And without these, we don't survive too well. This is really the entire universe inside our body. Together, this defines self. And by defining self, now we can shoot missiles against the things that are invading, that are not good, and not causing collateral damage to our own tissue. That this regulation or dysfunctions of the immune systems precipitated disease like Alzheimer's, not Alzheimer's, autoimmune disease, because now your immune system is out of whack. You start having problem regulating things. So it's really the purpose of the immune system to preserve yourself and destroy, anything else that you define as non-self. Right. So going back to the protein, and all these things are done by peptides. So it remembers the COVID-19 variant as peptide, this 15 amino acid peptides. And we remember a lot of them. In your immune memory, it's nothing but category after categories of these 15 amino acid proteins that defines whether it's bacteria or parasites or fungi. It's loaded with these memories, right? So I want to impress you, how does the B and T cells remember things? And these peptides are really in your library. And it's managed, again, by barcodes. So going to this story, it's a special handshake, right, between cells. And if you go to the videos to the FDA meeting, all of them explain all these things in details. So if you look at it and you say, well, Herb never told me about this. What is this? I'm telling you now, this is a secret handshake. So the cells against the 3A, 3B, and 3Cs, right? I'm not adding new things to it. This is the immune system activating cells that would directly kill infected cells. These are antibody producing cells, the B cells. And the story I want to tell you, it's in the middle. And these are the things that linking the peptide, the 15 amino acid peptide in the middle. I want you to remember hot dog buns. The immune memory is remembering a lot of different hot dog buns that recognize these peptides. If you have a new infection tomorrow with COVID, probably you have another new 15 amino acid peptides, and then you have another set of cell surface receptors that recognize this, one on the T cells, one on the dendritic cells. So barcodes, all right? The barcodes that regulate how these proteins are folding. So one barcode will be like this, the other barcode will be slightly twisting the protein, there'll be another barcode, and there are many, many of these barcodes. How many? Unlimited. This is the cute part of your immune system, has unlimited capability to generate barcodes for this kind of thing. So you have unlimited memory for things. Now, I don't want to go into this, but this particular molecule called MHC, if I tell you what it is, you know what that is. If you know of anyone that have organ transplantation, the first thing they'll say, well, why do you have to wait for so long for your organ? Because you have to match the donor and the recipient's MHC molecule. It's called HLA antigen. And this particular barcode guarantee that every single individual on Earth carry a different set of this MHC barcode. No two persons are the same, except for identical twin. I have a set of fraternal twin, and even them will have slight difference in this barcode. And identical twins, it will be the same, but the expressions of the gene products will be different depending on their environmental interaction. All right, so these are hot dog buns. Each barcodes carry different hot dog buns, and these are hot dog buns, and then these hot dog buns will then carry into your immune memories, into more memory T and B cells, and then it directs things happening to control any kind of things in your body. So really, to me, it's a testimony that it is an intelligent design built into these, ever-changing hot dog buns to handle all kind of problems that we're going to face. It's astronomical number. Can you imagine, how much stuff that, in our young 100 years on Earth, we have to come across all kinds of things. All right. Now this is the last slide. Oh, this is important. The neutralizing antibody. So someone is saying, well, how long is it going to last? And you heard that 6 months. So you are scheduled to have the antibody once it's made. The antibody should go away pretty fast. You have to. Think about how the immune system works. It's really a volcano. You blow it up, you blow up all the bad stuff. But when you no longer need, when the pathogens are no longer there, when the pathology is no longer there, you don't want the volcano to keep spilling lava. You want it to go quiet. So the antibody is the first one to disappear. The cytotoxic T cells will stay a little longer. And then it raised an army of these memory cells so that when my niece, it so happened that she come against a variant again, her memory cells will just spring up and then start working from day one. You don't have to wait for 2-3 weeks before the immune system to wake up again. That's why the first prime, it takes so long. The second boost of the vaccines, your memory cells are already there. And not only that, every successive memory cells will give you sharper and sharper laser focus to the target you want to eliminate. One of the comments here is there are tests for antibody, but how come you're telling me about these T cells? How come nobody can test the T cells? Yeah, you cannot test them. These techniques require special laboratories to handle. So there are no commercial laboratories to do this. So don't even bother to ask. So this is my last slide. So the immune systems elegant as it is in equilibriums. Once this COVID things go away, we're in equilibriums with the virus because now we have firewalls, isolating all the infected host, and we keep an equilibrium. But what's going to upset this equilibrium and precipitate the next pandemic, right, is this. We now know there are shifting weather patterns. We kill forests. Now we're moving closer to a cave where you have a swarm of bats living next to you. The climate changes, food supply changes, and pushing viruses, bats, mosquitoes, in a very different migration pattern. The case in point, the West Nile virus. In 2001, in 2000, we barely started with the northeast corner of the United States. Within 3 years, the mosquitoes carrying the viruses and the birds carrying the viruses are found in California, right? So there are real world consequences to climate shift and pushing these reservoirs closer and closer to us. Now you may laugh at this. The human behavior it's in the chain here. We pay someone in the world's paying a lot of money for food for pangolin. Some raise civet cats because you can make coffee beans. I don't know whether you know it. Coffee Louettes, you can buy a can of coffee Louettes, $40 an ounce. Have you purchased expensive coffee? Go on Amazon. You can buy coffee Louette. It's the most expensive coffee in the world. And because Jack Nicholson's in the movie Bucket List, make it popular. And if you go to fancy hotels, you can buy $100 cup of coffee Louette. We talk about mink. I thought the mink trade was dead, but I didn't know that they're still alive and well. You can buy a bag of guano, the bat guano. So someone is going to the bat cave and buying these things. Why? We have compost, but someone said, well, you put these things in your compost, you make much better fertilizer. All right? So we need to really modify our behavior, how we interact with the reservoir, with these vectors. And the last thing is intense farming. We saw the devastation with the mink farm, right? In the world of vaccines, the human vaccines stay within humans. The avian vaccines stay within birds. Now it will both the human and the avian vaccines will infect swine. Inside pig, if you have all these commingle with different viruses, that's how recombination occurs. And that's why new species come about. We really cannot, we have to think about a better way of doing farming, all right? So these are the pressures that's forcing the increased transmission rates, replication rates, mutation rate. And the only way now we're in the situation, the only way we can cut them off is to vaccinate, mass vaccinate the world, and so that we now cut off the virus. Okay, so I'm going to rest my case. Oh shoot! There are other stuff. Are you going to get infertile? No. The side effect of egg protein. There's no egg proteins in here. I don't need the vaccines after recover from COVID. No, you need it because you may be dealing with new variants because the vaccines only give you 90 days of antibody reserve. Vaccines have tracking device. I don't even want to go there. The vaccine will give me a positive COVID-19 antibody test. Yes. Antigen test, only a brief period of time, but certainly should not give you a positive test for the PCR version. I don't want to wear a mask, but you may not be infected, but you may be carrying the virus to other people who don't have the strong immunity. So I would say still practice. All right. I'm sorry I went over, but I'm going to give the time back to David. Any questions I can entertain? Feel free to send me questions through this email. Also, David knows how to get in touch with me. David, it's all yours.
Thank you, Dr. Chow.
Please, Herb.
I forgot to tell everybody we're going to have an examination now. Now they're all running away. No, it's really, really good, and really, I learned a lot personally, and I didn't expect this kind of level of graduate courses about vaccine. So thank you, Dr. Chow. There are 3 questions for the sake of time. I just mentioned maybe 2 or 3. One is the question about immune escape. Are you suggesting that we should not get a weaker effective vaccine like Sinovaccine, or Sinovaccine tend to have more potential to develop immune escape since it's not as effective as the other?
Yeah, if we want to control the virus, we really need to shut down the current variants. And you know, the Moderna and the Pfizer and the J&J, I see the potential. Just go after the spike and pull down all the variants that are existing. Now, you know, short of doing that, you're going to leave some variants untouched. This is very much the ideas behind bacteria resistance, right? You have antibiotics you give to the patients. You only eliminate 90% of the bacteria in that disease. The remaining 10% now learn how to be resistant. And that's how the bacteria resistance story started, you know, many years ago. Here, we don't call it bacteria resistance. We call it immune escape. But, you know, the underlying mechanisms are very similar.
Okay, thank you. Second is some vaccine are DNA-based, vector-based, that can enter into the nucleus and come out. So you seems to suggest to reassure us that the effectiveness is the same, and as in there should be no concern for our DNA interruption to our DNA, correct?
Yes, at least for the few vaccines that are now FDA approved. AstraZeneca that is about to come in April. So at least those 4 vaccines, you don't have to worry about it.
Better. Okay, thank you. This is a question. What is a long-term? What is a possible long-term negative side effect that might not be known for years to come? Are there?
Well, just like, you know, all of us have numerous vaccines given to us, you know, since I just got shingle vaccines. I really don't know, are there any long-term side effects that will come out and bite me when I get to 95? I can't ask that question because I don't think anyone can tell you. Now, if you have allergy to some of these products, yeah, I mean, it may be long-lasting if it cannot be corrected right away, but nothing that I can see that it's long-term, it will negatively impact your daily life, or it will precipitate other diseases in your life just because you get this vaccine. But, you know, it's a question that I really don't know. I don't think anyone can answer that.
Okay. Someone who has thrombocytopenia, can you comment on a vaccine that affects existing autoimmune deficiency?
If you have you said
thrombocytopenia, which means that the decrease in platelet.
Yeah. Remember the case in Florida, the doctors got on the vaccine trials, and he ended up with ITP, which is the thrombocytopenia, and he ended up dying of a bleeding episode. the incidence is very low. So if you scour through the database, you don't see enough incidents to cause alarm. Maybe there are risk factors that we don't know why certain people after vaccines have blood clot or have thrombocytopenia. I cannot tell you, but the only thing, for example, the AstraZeneca, you see the logic here. Well, we see people with blood clot, but the incidence of blood clot is not any higher than the non-vaccinated individual, right? So you cannot say that the vaccine is causing it. Bell's palsy is another incident. When it first came out, we hear in the news that 4 or 5 people getting Bell's palsy, it must be bad. But then if you look at it, you get flu vaccines, people have Bell's palsy. Well, it's not the vaccines causing Bell's palsy. In a community, on any given time period, you will have people come down with Bell's palsy. It has nothing to do with vaccine, right? So, what are these causal relationships? It's really difficult to understand. For 2 years, I sat on the International Vaccine Safety Committee. I'm very conscious, conservative when it comes to vaccines, because I've seen a lot of really direct causal relationship with vaccines. There are those examples, but doesn't seem like this vaccines fit those categories. That's all I can say.
Oh, thank you. Well, I think one more question related to a prior question regarding immune escape, which is basically a virus term for bacterial resistance. Do you say that there will be concern if we have a massive vaccination against COVID-19 worldwide, that the immune escape might occur? I think you have answered that in the previous question, but can you say that? Can you talk about it again?
There is a community of scientists. I think it must be either French or Belgian immunologist. He's the great proponent that's saying that you either have to give a strong vaccines that can pull down all the viruses to shut off. If you give a not so good vaccines, then you're going to create the situations where we just talk about, because now just think about it. Let's say you have a situation, you have 3 populations of viruses in your body, right? One has, 2 of them can be controlled by the vaccines. You create antibody and you killed all those. 1 populations have escaped. That populations will probably start growing in that person's and get passed on to the next person's, and then you start a nucleus of infection. So, I think that's what that question's alluded to, is you really need to have not only strong vaccines, but broad vaccines, not only tackle the spike protein, but tackle the nuclear proteins, the membrane protein, the sugar molecules on the protein. So now we have multiple arms to shut down the virus instead of just the spike. Because if the spike doesn't work, kind of like, if I'm drowning in the ocean, don't tell me you throw 1 buoy to me. Why don't you throw, a plastic bag with air, a floating buoy, a wooden oar, help me with anything. So develop a broad immune reaction with a broad vaccine has that advantage of have multiple arms of holding on to different population of virus to shut them down. And if you, short of doing that, you may risk of having certain population of virus just stick around. And then when time comes, it breaks the lock again, and then start having another outbreak. You look at HIV, it's like that in real life.
Thank you, Dr. Herbert. I think there's one more last question, and we should not take this too long. What is the chance of reinfection after I recover from first infection?
Well, reinfection number is not very high, but it does exist. There are multiple cases, not very high. So I would say, no cause for concern. But if you think about the immune escape scenario, it's it's possible. But what I also want to say is this. For example, my niece, right? She now in her bodies, even though, well, in her bodies, she has the advantage of broad vaccines, in the form of real virus. So not only she has antibody to the spike, she has different population of cells try to shut down the nuclear proteins, the membrane proteins, and that helps. But if you don't have that kind of broad spectrum profile, then you have to worry about it, because the residual viruses could give you a bad headache if you come across it. But having said that, go back to the immune memory story I just showed you. So let's say you get vaccinated, you only have one populations of anti-spike cells, T cells and B cells. Let's say they all remember spike because they have never seen anything else. Now the variant comes in, all of a sudden you get sick, but you still have memory of the virus, not very good, fuzzy. You may get, you won't get really sick. You may get a little bit sick, but after that round of mild sickness, you now have the full capacity of the viral proteins, the membranes, the nuclear capsids, the spike proteins, the real things after that variant attack, right? So I would say don't worry about it. Reinfections will occur, but it should be mild and shouldn't be as bad as, as if you are exposing to the virus the first time. And then after that round, then you get exposed to the real thing, so you'll be good.
Thank you, Dr. Chow. I think for the sake of time, we're not going to continue further question, but this talk will be is recorded, and if you want a recording, please send me an email. If you have further question and you don't remember Dr. Chow's email, you can send me an email and I will pass on to him. And so next week we'll have Dr. Chow come back to talk to us again on the other topic. And
not next week, 3 weeks from now.
Oh yeah, next month. So let's close in prayer, and thank you very much. Dear Lord, thank you so much for what we have heard today. And the most beautiful thing is we begin to see how wonderful our body is made. And this is impossible except you created us. And we thank you for the opportunity to live here, to be able to get vaccine, to be able to have opportunity to serve you after having a healthy body. And thank you for the talk tonight that I pray for those who heard this talk can get more educated and decide whether to get a vaccine or not, can weigh their risk versus benefit. We thank you, Lord, for you are our God and our Savior. In Jesus' name we pray. Amen. Okay.
Thank you,
Thank you,
Herbert.
everybody.
Thank you.
Thank you, Uncle
Wonderful
Herbert.
meeting. Thank you.
Right.
Thank
Bye-bye.
you.
Thank you.
Thank you.
Thank you.
Thank you.
Opened your mic to say thank you.
Thank you.
Thank you, everybody.
Thank you.
I think chat is recorded.
So that's
Thank you.
How do you get the
Thank you.
recording?
It's already recorded. We ended this.
night.
Hi.
Night.
How do you?
Good night.
It's already recorded. We ended. It's recorded.
Okay.
So what did I miss? I went to the bathroom.